Right. Hi, I'm Uroosa Ejaz. I'm working as a lecturer at Fabrication University, Karachi, Pakistan. I am a PhD scholar in Department of Microbiology, University of Karachi. I'm very thankful to the Magnus Group for providing me this opportunity to present my research project. The topic which I am presenting today is fabrication of methylcellulose polyvinyl alcohol as porous composite. Next slide, please. Okay, halophytes plants. Halophytes are the plants which have the ability to survive in saline environment. Next. Okay, so what are the saline soils? In our coastal areas or marshy areas, the soil which is present contain different types of salts. They mainly consist large amount of sodium ion. Sodium adsorption on clay particles also reduce calcium and magnesium content of the soil. The marshy soils are typically low in oxygen amount. They are high in carbon dioxide and they contain high amount of methane. And the composition of those marshy soils keep changing because of the flow of the tides. Next slide. So if we talk about our planet, the 80% of our Earth is covered by the saline water. So it means we have a huge diversity of halophytic plants. The plants which can survive in saline environments are termed as halophytes. And this term was first coined by Pallas in early 19th century. Next, please. Okay, so the term halophyte has not been previously described in the literature, but now we have its proper definition. In 1903, halophyte plants are considered which have the ability to grow in saline environment, and they can also survive in unordinary soil condition. In 1928, this definition got revised, and now the halophytic plants are those which can tolerate a high salt concentration. In 1957, again, the definition was revised, and the plants which grow exclusively on salty soil are considered as halophytic plants. Next, please. So according to the salt-loving condition, there are three categories of halophytes: intolerant, facultative, or obligate. Intolerant plants are basically those plants which cannot tolerate the salt amount in soil. Facultative are those which can grow in salty environment, and obligate halophytic plants are those which require high amount of soil in order to grow. Next, please. So if we divide our plants into two categories, glycophyte and the halophyte, so if we increase the salinity, as you can see here in the x-axis, the growth of the glycophyte will get reduced because they can't survive in salty environment. But if we talk about the halophyte plants, the obligate halophyte plants can grow maximum in the salty environment. Next, please. So there are many species of halophytic plants. If we talk about only about the Pakistan, there are around 450 halophyte plant present. Among them, Phragmites carca is one of the halophytic plant which can grow in salty environment. It is herbaceous woody clump and it is mainly composed of cellulose, hemicellulose, and lignin. Next, please. So the aim of my research project was to extract different components of Phragmites carca, especially the cellulose, Then the conversion of cellulose into methylcellulose. After this, the use of methylcellulose to form a porous composite. Next, please. All right, so methodology start with the collection of halophytic plants. The Phragmites Karka plant was collected from University of Karachi, situated in Karachi, Pakistan. The plant was checked by Dr. Salman, who is director of Halophyte department. Next, please. Okay, after the collection, the plant material was ground to 100 micron size, because the more reduced the size of the plant biomass, the more reactive it will become. So after the grinding, the plant was treated with the boiling water in order to remove water-soluble polysaccharide. After this treatment, the content was filtered. The filtered was saved as a content of water-soluble polysaccharide, whereas the residues were again treated with the HCl. After the acidic treatment, the plant material was again filtered. The residues were further treated by sodium hydroxide in order to remove the lignin content. And after the removal of lignin content, the residues which were left out were considered as cellulosic content. Next, please. Okay, so after the extraction of cellulose, we have to methylate it, because the cellulose is water insoluble polymer. So in order to make it water soluble, we have to attach methyl group on its surface. So for the methylation purpose, dimethyl sulfate was used. Initially, the extracted cellulose was mercerized by using sodium hydroxide, and then DMS, dimethyl sulfate, was added in order to perform the methylation. Next, please. Okay, once the methylation was completed, the methyl cellulose was soaked in the distilled water, and then it was get stirred. After the stirring, the polyvinyl alcohol, which is again a biopolymer, was mixed with the methyl cellulose solution, and the polymer solution which was formed was then lyophilized. Next, please. For the analysis of the extracted samples of Phragmites carga, scanning electron microscopy was performed at University of Karachi. Next, please. The composite which was formed and the extracted byproduct of the process, like lignin, hemicellulose, were tested. Either they have antibacterial property or not. For the antibacterial testing, 3 and microbial cultures, Staphylococcus aureus, Escherichia coli, and Klebsiella pneumoniae ATCC strains were used. Next, please. Okay, so the extraction process resulted in different components: lignin, hemicellulose, cellulose, and water-soluble polysaccharide. And we found out that the Phragmites carca contains 10% lignin, 43.5% hemicellulose, 24% cellulose, and 5% water-soluble polysaccharides. Next, please. Yeah, so here you can see the pictorial representation of the extracted component. You can see the water-soluble pectin, acid-soluble pectin, the hemicellulosic content, the lignin content, the methyl cellulose, and the composite. Composite is basically the biopolymer mixture of polyvinyl alcohol and methyl cellulose. Next, please. Okay, whenever we perform the methylation of cellulosic component, we have to calculate its degree of substitution. So the cellulose structure contains 3 hydroxyl group. So it means we can replace these 3 hydroxyl group by the methyl group. So maximum degree of substitution could be 3. In our project, we obtained 3 value, which showed us that the maximum methylcellulose group, the maximum methyl group get attached with the cellulose. Next, please. Yeah, here you can see the SEM images of extracted components. In the first image, you can see the native Phragmites carca. The structure was quite rough. After the water-soluble extraction, here you can see water-soluble pectin and acid-soluble pectin. The acid-soluble pectin quite have uniform or smooth, silky-like appearance. Next, please. Okay, so here you can see the SEM images of hemicellulose, lignin, and the methylcellulose. So the structure of the lignin was quite rough, whereas if you compare it with the methylcellulose, the structure becomes quite smooth. Because the most of the lignin part, the most of the hemicellulosic part, water-soluble polysaccharide are not present in the methylcellulose. So that's why it is now more pure and the structure appeared quite smooth and silky. Next, please. Okay, so this is the very important image. This is the composite SEM image. Here you can see the cellulosic nanofiber structure. The needle-like structure which are shown here are basically the cellulosic nanofiber. It means that we successfully converted our extracted cellulose into the methyl cellulose, which can serve as a nanofiber. Next, please. Okay, when we did antibacterial testing, we found out that the extracted component of Phragmites carca possesses antibacterial activity against 3 pathogenic strains: E. coli, Klebsiella pneumoniae, and Astorias. It shows us that the byproduct of the process can also have many important applications like antibacterial activity. Next, please. All right, so from this research project, we can conclude that halophytes are those plants which can be used as a raw material to obtain multiple products. Like in this study, we obtain lignin, hemicellulose, polysaccharide, and the cellulose. The extracted cellulose can be converted into the methyl cellulose, and then that methyl cellulose can be used to form a porous composite. The porous composite, which we formed in this study, can be used in biomedical devices. It can be used as a drug delivery system. So there is a wide range of biomedical applications. Next, please. This research project was presented in many national and international platforms like 7th All Pakistan Dice competition. Next, please. This project also received prize money in the ZAB E5 competition, which was held in Zabis University, Karachi, Pakistan. Next, please. We presented this research project in Sindh Higher Education Commission Research and Technology Showcase 2022, which was conducted at Karachi last year. Next, please. It was also presented at Faisalabad University. Next, please. So if you want to know our team, I am the Uroosa Ejaz, the supervisor of this project, Dr. Mohammad Suhail, who is associate professor at Karachi University. Mr. Ejaz Ahmed, Mr. Danial, and Mr. Tamiz Ahmed are my students which worked on this research project. They did their BS from Zabis University. Next, please. So with this, I want to thank you for all the listeners, all the participants, and the Magnus Group, the organizer, for providing me this opportunity. Thank you.